Biological Dynamics ACE system can selectively isolate, quantify, and analyze necrotic biomarkers directly from blood. When cells undergo necrosis, they release their contents, large partially degraded molecules, into their surrounding environment. The immune system cleans up this debris by breaking down the particles and removing them from the bloodstream. This natural process is typically completed within 24 hours. With critical diseases such as cancer, the chaotic growth of the tumor causes chronic necrotic cell death in the tumor as well as in the surrounding tissue. This debris is continuously released into the bloodstream, and the natural immune response is unable to effectively remove it all. This necrotic material, if selectively isolated, becomes a very important biomarker to track tumor burden and response to therapy. Current sample extraction methodologies obscure these relevant biomarkers due to cumbersome and destructive processing. The necrotic biomarkers become a needle in the haystack, and the ability to track them becomes extremely difficult. With the ACE platform, however, these necrotic biomarkers are selectively captured, even in the presence of wild-type background material. The ACE system uses an AC electrokinetic technique to rapidly capture cell-free DNA in the form of oligonucleosomes, RNA trapped in exosomes, large aggregated protein biomarkers, and other cellular debris directly from a blood sample. Once isolated, these biomarkers can be quantified to identify the overall concentration or analyzed further to track specific mutations or proteins within the debris. This rapid characterization enables streamlined, individualized treatment for a wide variety of diseases.